X-ray crystallography has contributed enormously to our understanding of biological molecules. It's difficult to understate its importance, but it doesn't tell you everything. And I think I'm rather going to be preaching to the choir here, but I've summarized the important factors on this first slide, if you phase out a bit later on. So the problem with X-rays is they're damaging, and they will perturb the redox states, and they don't show hydrogens. So understanding how biological molecules work is challenging with that lack of information. Neutron crystallography, on the other hand, doesn't cause noticeable damage. It means we can do structures at room temperature, or physiological temperatures. We don't get photoreduction, so we don't perturb the redox states, and it shows hydrogen positions, which not only is essential for understanding the chemistry of biochemical reactions, but we can understand charge states, binding, hydrogen bondings, how drugs bind, etc., etc. And we can also do some interesting stuff on showing exchangeable sites. There are disadvantages of doing neutron crystallography. We need large crystals. We have to do some hydrogen deuterium exchange or perdeuterate the samples. Data collection is very slow compared to X-rays, and the number of facilities is extremely limited. So LMX should enable access to collect data locally for the UK community. It'll increase capacity. It will also facilitate access to other facilities such as ESS in the future. So in order to understand biochemical reactions, we need to be informed about all the atoms, including hydrogen, and they're difficult to find. There are several reasons for this. One is you need extremely high resolution in order to separate something that's in the order of an angstrom away from another atom. The number of electrons that a hydrogen atom has varies from 1 to rather less than 1, depending on what it's doing, because the scattering is proportional to the number of electrons. And the peak of electron density is not going to correspond to the atomic position. Of course, a proton has no electrons. And even with sub-angstrom resolution data, and this is mine, so I'm not criticizing anybody else, you don't always see things that you should see. We know there are hydrogens at these positions, and the difference map should show those hydrogens, but it doesn't. So the other thing is, it's like censoring all the important bits, the interesting bits, shall we say. So the bits that you want to see are censored. And so what you end up doing is guessing these interesting bits, and in this case, the hydrogens. So you wouldn't try and work out how a protein structure an enzyme works from an alpha carbon trace. It may provide you with a pretty picture, but it's not going to tell you how it works. You need the positions of hydrogens to do this. And the power of neutrons is to distinguish isotopes, show protonation states, and it allows you to properly see hydrogen bonds. You can see how a water works. In X-ray, you just get a sphere from the oxygen, whereas with neutron, you get this sort of banana shape where you can actually see what the hydrogen bonds are doing. And you can see if waters are spinning as well. You can also distinguish oxygens from nitrogens in side chains. See the amide flip, because the scattering is from the nuclei and it's different. This isn't just an academic pursuit, of course, it's very important. This is a paper from Andrei Kovalevsky's lab recently, whereby by doing room temperature structures of the SARS COVID-19 protease, you can see the protonation states of the ionizable residues. You can see how the enzyme works from this, and because you can do it at room temperature, you can see what these protonation states are at room temperature, which is a, you know, if you've ever, as a biologist, if you've ever made a tris buffer, you know how dependent on temperature the pH of that buffer is going to be. Your ionization states do change with temperature, and you can find out really what's going. So this is, you know, it gives you real information. If you want to design a drug, you want to inhibit something as unpleasant as the SARS COVID-19 protease, this gives you the information to do it. And my interests are in redox enzymology, and the X-rays have about 10 to the 3 times the energy of the first ionization energy of in general of elements. And so if you bring in a whole load of intense X-rays, you're going to cause all sorts of things. You've got a lot of water in there apart from just the protein. Most of the X-rays are going to go in, are not going to be scattered in your coherent scattering. They're going to be, many of them are going to cause damage, and they're going to perturb the system. So if you've got a redox system, you're going to perturb it, and you're going to do general damage as well. A lot of electrons are going to be released. You can see this with, for instance, in flavoenzymes, you can see the flat flavin ring will will bend into the reduced form if you give it enough X-rays, and you're seriously cooking these things. So as a synchrotron, with the amount of photons going in, you are cooking samples, so you've got to freeze them. So one of the things that we've been working on has been the heme peroxidases, and we wanted to know, to cut a long story very short, is what the state of these intermediates were. Was it iron oxygen or an iron OH in these intermediate states? And by using neutron diffraction, we could tell what was going on. We could track these intermediates and we could see what states they were. It wasn't possible to infer this from bond length for various reasons, but photoreduction is amongst those. And we also came up with some unexpected findings about the ionization state of this histidine here in the active site. So a lot of chemistry of these enzymes had been guessed, because that's all the information we had, but we were able to end up with a lot more solid information. Another thing we found when looking at the proton transport mechanism, the sort of Grotus pathway from the substrate here through to the ferrule center, we found that there's an arginine that has its pK effectively perturbed, so it becomes a guanidine rather than a guanidinium, which is extremely unexpected, shall we say. We have a problem though with sample preparation. As I said, we need large crystals and we need to perdeutrate. So we get some interesting effects with our maps. Because the hydrogen has a negative scattering length, we end up with a cancellation effects. So with exchanged samples, we end up with this sort of cancellation here to compare to the X-ray here. But if we perdeutrate our samples, we can get these lovely pictures which show where all the hydrogens are, even at relatively modest resolution. Now, obviously, anybody who's interested in protein structure should really be doing neutron crystallography, but I asked the LADI and BioDiff about their usage statistics, and they're oversubscribed. And I'm on the panel for LADI and for Oak Ridge's user panel, and they are heavily oversubscribed. So a lot of good work doesn't get done because people can't get access to the equipment. if we set up LMX here, it'll be at the hub of macromolecular crystallography in the UK. Not only macromolecular crystallography, but also structural biology with the microscopes, the electron microscopes and everything else like that. So LMX here will be a superb facility to have, because there's development going on all the time at Diamond with the dials people to tell you how to work out how to best integrate data. We've got the central laser facilities. We've got all the work going on at the Rosalind Franklin Institute. It's a hub environment, and this is something that we could maybe talk about a little bit more later on. Now, very briefly, the way you can collect data is either on a image plate device, such as at LADI and DALI at ILL, or BioDiff. BioDiff is monochromatic, and the quasi-Lowry technique is used at LADI and DALI. I haven't got time to go into those in detail now, but we can discuss those more in detail. And up and coming, there are spallation sources. Now, there is availability at Mandy, but it's in the United States, and you've got to get over there, and they're not going to pay for you to do that. There'll be ESS coming online eventually. There's IBIX, and we're hoping LMX will come along here. Now, this is an importantly different technique because it uses time of flight and allows you to get a lot of information really very quickly. And so LMX will be here. When I say here, there, where it's going to allow state-of-the-art macromolecular time of flight neutron crystallography. And this will help us if we want to go and use the really big sources like ESS in the future. We can do important work here. Now, the instrument is cutting edge technology. It'll surround the sample, and it will and it's going to use these wavelength shift fibers, whereby the scintillant combined with a fluorescent thing, a fluorescent compound, allows you to get light going into fiber optics. And by having double layers and having time delay and suitable electronics on there, you can locate the events. And the team, who I'm not at all involved with, so have developed this enormously, and they've got, this is the prototype being tried out at the what I'll call a small molecule station at the moment at ISIS. But this new double layer technology is going to enable very fine effective pixel sizes, and going to be very, very efficient. This is the sort of wavelength we're talking about, around the 2.5 angstrom is what we would probably want to be using for, this is the band, if you like, because of course we're going to be using a large band of wavelengths. And this is much, much more efficient than the current detectors. And fine pixel sizes, this will also, effective pixel sizes, which will allow the large unit cells that we need for macromolecular crystallography. This is an example from a small molecule, the sort of gain that we can get. And it's a chart of the comparison of the new generation of detector. And you can see, so it's a huge leap forward in terms of what we'll be actually able to do and how we'll be able to collect data. Just to compare, the LADI that we would be the instrument of choice at the moment for collecting data with an image plate, the technology is completely different. LADI has the disadvantage that you're collecting all the wavelengths at once, and so you get a lot more noise. Now, you know, I could have spent an hour telling you about the advantages of this, and I could have spent another hour telling you about why protein crystals need to be done with neutrons. If you're further reading, though, there's an article by Flora Muller from Oak Ridge in The Biochemist, which is open access and easily found on Google. Or if you want something a little bit more advanced, there's a copy of this Neutron Crystallography and Structural Biology, Methods and Enzymology volume that came out last year. And that